Glad to be here. And yeah, so I guess April 2022 was the last time we were out. We were recently funded at that point. I think it took us, one thing that took us a bit longer than we had hoped was recruiting students, right? So part of our mission at LSU is, of course, training other PhD students, master's students, people that are going to work in the field. And that takes time to recruit them, especially like post-pandemic. So I think it took us about a year to get 2 students in place. So we didn't start our field sampling and research activities until February of this year. As you mentioned, this is funded by the US EPA through the Gulf of Mexico program. So they put out a call specifically looking at improving community health through microbial source tracking. And they really asked for 2 things. 1, a microbial source tracking component to the project, and 2, like an innovative demonstration as a component of the project that would impact human health. And so I'm doing the microbial source tracking. Sam is leading the UV disinfection, and then our other partner is Dr. Brady Skaggs, the Contamination Conservancy, and of course Ronnie Carter, who we work with real closely on the field sampling, and he's an OSS on-site sanitation expert, so we work closely with them to learn about those systems. And then we've all appreciated the support of DEQ and the Tangipahoa Parish Council. This, when it comes to human health, right, this is a picture actually we took during our field sampling. So these are children actually swimming. This is in the Albany River. So this is one of our field sampling sites. And I guess the point I'm trying to make by putting this picture here is this isn't like just a theoretical exercise, right? People are actually in contact with these rivers, especially children. And so the microbes living in these places can have an impact on human health through exposures such as swimming, boating, fishing. One thing I just need to note here is this footnote says, right, that although we're funded by the EPA, this presentation is just our experience and opinion as experts. And it's hasn't been reviewed by the EPA, and it's not their opinion. So just a review on the project scope. We're testing 4 sites on the Yellow Water, pretty much beginning north of Hammond and coming down toward Ponchatoula, and then 4 sites on the Albany. I think we cover, I can't remember how many river miles we cover, maybe like 25 or so. It's a long way. So we're sampling at these 4 sites monthly for 18 months. And the reason we're doing it this way, we're kind of trying to understand how the water quality changes as the rivers move through these developed areas. So we're doing that, and then we've also got the UV chlorine disinfection, which Dr. Snow will talk about in a moment. And then on the community engagement side, of course, part of why we're here is to engage with the community. We're out once a month doing our field sampling, and we're always looking for opportunities to meet folks in the community and try to see what the problems are, see how we can help. So that's another component of the project. We began the field sampling in February, as I mentioned. We just finished our 8th trip last week, so next month will be halfway. And we have a team of students working on this. We've got 2 master's students and a PhD student that help us with our field sampling. And during our field sampling, we measure a variety of water quality parameters: dissolved oxygen, temperature, oxidation reduction potential, conductivity, turbidity. And then we also measure some microorganisms using standard methods, EPA standard methods, so nitric oxide, total coliforms, and E. coli. And this really, I'm going to show you some data tonight, and it's only 8 data points times 4, so 32 points per graph. It doesn't seem like much, but it is a ton of work that goes into producing these data. We come and collect the samples typically on Monday. We usually leave LSU at 8 a.m. or 7 a.m. We drive out here, collect our samples, have lunch, drive back. So we finish that day about 3 p.m. and then we spend the next 4 days analyzing the samples. So it's really a week worth of work. And So thankful to our students who, put in a lot of hard work and a lot of hard effort. You can see Ronnie here helping us out along the way, making sure we don't screw up. I do kind of want to mention here
watching for trains.
Yeah, train spotter. I do want to mention, of course, LSU, we're a land grant public university, so an important part of our mission is educating our students. So I've got 6 undergrads working in my lab, all of which have touched these samples and work on them. And this is an important part of them getting practical on, hands-on experience on how do we measure water quality, why do these issues matter, how is this affecting the local community. So I would like to just acknowledge the role that students play in getting this work done, and the privilege it is to get to help teach them some things. There's a lot of data I could put up here. I've put turbidity just kind of as an example. We've got on the left-hand side turbidity over time. So for each of these rivers, we've got 4 sites. So we've got yellow water 1, 2, 3, 4. So 1 is upstream and 4 is the farthest downstream. And we've started in February through September. And I mean, we don't see anything that's particularly outside the realm of normal for a river. This was a rainfall event here where we got some high turbidity based on what was washing into the river. Here recently, we've definitely run into some probably mining runoff. So you can see this is our last, I actually got the data in here from our last sampling where we saw, especially in the Natalbany, way up in the Natalbany River 1, really high turbidity. But you can see these two are both rainfall event driven. So we can look at this over time, and then we can look at just how the distributions are at each site clustered together. And you can see that we definitely have some kind of extreme events here. Now, I study microorganisms, and one of the things that's kind of difficult about microorganisms is they don't necessarily really correlate with these other water quality parameters. You can go out and the river can look perfectly clean, pristine. Let's jump in and go for a swim, and there could be microbial contamination. On the flip side, it can be really turbid. There's not, public health relevant contamination. So sometimes it's hard to know just based on this kind of data what you're looking at from a microbial perspective. So some of our microbial counts, I'm comparing, this is historic data, so we pulled this from the EQ's database, and here in Louisiana, fecal coliforms are kind of what we use to assess water quality. As part of our project, we're looking at E. coli, so this is us, this is our data, total coliforms and enterococci, mainly because when we wrote our proposal, we were kind of trying to fit into an EPA frame, and most of their primary contact recreation standards are based on E. coli and enterococci. So I think, if we just look in general, I don't think we see anything that really deviates from this historical norms. Our total coliform counts, so a lot of our total coliforms are what we call too numerous to count. So there's so many that when we try to count the individual colonies, we can't do it. However, coliforms include a lot of bacteria that are free living, especially in subtropical climates. So there's a lot of bacteria in here that might not be associated with feces. They're just free living coliforms. As we come down to the next level, we get the fecal coliforms, right? So it kind of makes sense. We're seeing total coliforms up here, pretty high, and then when we drop down to the historic data for fecal coliforms, a little lower. Then when we move to our E. coli data, it's a little lower because these things are like subsets of one another. And so we would expect the counts to get lower as we get more specific. And really, the E. coli and enterococci are the kind of the human health, from an EPA perspective, relevant ones. In general, we see higher counts in the yellow water than the Natalbany. I don't think that's probably that surprising, given the areas those rivers are flowing through. So the real component of this project that's kind of still under development is the microbial source tracking. So one of the limitations of these bacteria, right, E. coli, all warm-blooded mammals shed E. coli in their feces. So when you find E. coli, that doesn't necessarily tell you very much about where this feces is coming from. Is it cows, raccoons, waterfowl, human sewage? And so the science that we use to try to figure that out is called microbial source tracking. And so it turns out that depending upon where the contamination comes from, there are unique genetic signatures. And we can look for those signatures, and based upon it, finding them or not, and the level, like how high the number is or low, we can start to kind of tease apart, okay, we have fecal contamination, and like we're seeing some markers that are associated with human sewage, we're seeing some markers that are associated with cows, and that tells us how to begin to mitigate the risk, right? Because, you know, like I worked on a project in South Carolina before I went to grad school. We were working in shellfish waters, and so there were some shellfish beds that were getting shut down due to fecal contamination. Everyone thought, okay, well, these are failing septic systems, that's the problem. We applied the microbial source tracking tools, and come to find out it was deer, raccoons, wildlife. And so what was actually driving this contamination was development was pushing these animals into the marshes where they were defecating, and then that material was flowing down the river to the shellfish harvesting beds. So you can imagine, right? Imagine if you had invested a bunch of money trying to fix septic systems, and then come to find out that's not even what's causing the problem. So that's why we use microbial source tracking as a way to try to understand where do we need to aim our solutions. So these assays take a long time to kind of start up and develop. It's really labor intensive. This is my PhD student, Shimul Ghosh. And so I've been training him on digital PCR techniques, which is what we're using. And so we spent the last few months kind of like stretching our legs, getting all this sorted out. And so one of the things that we're doing, one of the things you're really interested in is your precision. So when you measure this, if you measure it repeatedly, do you get the same number over and over? Are you getting a lot of variation in your measurements? And so what we've been doing is using control materials, making sure our instruments are working correctly. We've been looking at the correlation between our technical replicates. So what we do is we'll measure something repeatedly, and in an ideal world, we should get the same number every time. But you know, there's human error in these measurements, right? We're pipetting, it's really small amounts of fluid. So the closer we get to 1, these R squared values, the closer we get to 1, the better the agreement is. And I would say we're doing pretty good. Some assays are better than others. These are three different assays we're using. And we basically have collected samples around Baton Rouge just to kind of test these methods. So we're kind of, long story short is we're getting these molecular methods kind of dialed in very nicely where we feel very confident in what we're doing, and then we're going to take the samples that we've frozen and start testing them. Other things we're up to, you mentioned the Louisiana Water Resources Research Institute. They gave us a little bit of funding to look at antibiotic-resistant E. coli. So we are now taking these samples and having to look for antibiotic-resistant E. coli in them. And then more recently, we're starting to develop something called passive sampling. So when we go out to the river, we just take our sample bottle and just dip it in. So what we get, right? Whatever's flowing by at that exact moment it's kind of a snapshot. But of course, snapshots can be deceiving, right? Depending upon what's going on. So we've started to develop this new technique called passive sampling. On the right here is a passive sampler. So that's a 3D printed sphere. It looks like a wiffle ball. It's got little holes in it. And what we do is we put a 5-gram packet of activated carbon inside of it. And activated carbon likes to basically grab things out of the water, things absorb to it. So we leave these in place for 4 or 5 days. As microbes flow by, they stick to that activated carbon, and then we're able to test for them. And it makes it way more sensitive than a grab sample. So now instead of getting like a snapshot, I'm getting kind of a like over time picture of what's flown past that sampler. So those are some of the other things that we're tinkering with and doing kind of at the same time as we're executing this project. So I'm going to turn it over to Dr. Snow to tell you about the UV LED work.
All right. So, here to give a little bit of an update, just kind of how the project is going. I can't tell you everything because part of what we're trying to do is develop new technology and patent and all that. On the other hand, I can't tell you super exciting things because we're not quite there yet. So, I'm going to give you what I can give you and kind of walk you along our process, where we are in that process. And generally, this challenge of these on-site treatment units, aerated treatment units, is kind of what we're after with this particular project. The idea is that we have a lot of these here, and they're not disinfected typically if it's an individual homeowner situation. And so that discharge out into the environment is then going to, maybe be a source for all these microbes that Dr. Bivens is tracking with his group. So it's this complementary approach. We hope to demonstrate this and perhaps even bring it to a new technology kind of a spot. So a little bit about these ATUs in the first place. Well, I mentioned they lack disinfection. So we want to try to fix that. That's a major goal of ours, is to see, can we put something out there that would be effective, both in terms of how much microbes we're killing, but also how much does it cost to maintain, to operate, and can it be something that is there in a more permanent manner that the homeowner wouldn't have to really think too much about. There's a couple of challenges outside of just that that are a little bit outside the scope here, but may become related. So if you're not operating one of these ATUs well, you're probably not removing the oxygen demanding substances. So then your discharge, you're basically not, if the treatment unit is not functioning properly, then the nature has to do that function. And so that spoils our environment, that's bad. So that's kind of related, and I think Pump Treatment Services has done a lot on trying to educate people how to operate and maintain these better. And we'll see also, again, with the turbidity, there's a...
Good evening, patrons. The library will close tonight at 8 o'clock p.m. All public computers will shut down for the evening at 7:45 p.m. Thank you.
All right. So the turbidity, and actually I should check. Okay. I'm not going to take 45 minutes. So turbidity is another one of these things that just simply operating, properly maintaining the aerator system. If it's not going properly, or if it's trying to get air and it's not getting much, maybe weeds growing around the pump or something, that can cause big problems for the treatment itself that our potential disinfection system can't handle, and will actually be bothered by in some sense. If we have a lot of turbidity coming out, if we're going to apply UV or chlorine or anything really, that gives bacteria a place to hide. So that's a potential challenge, a little bit outside of our scope, but one thing that maybe we can look at is if we were to install something that had a little detector in there for turbidity, maybe that could alert the user to say, hey, we have some trouble here, please do some maintenance. Okay, so then the other thing as we think about where we would put a disinfection system, just a couple of thoughts. We could put it right at the outlet, but then you have to dig up the system itself and do some excavation and all that. The alternative would be to put it at the very tail end of your system, but then you either have to put some electrical wire out there to power it, if it's maybe use a solar system. So there's a few different things like that. We're just starting to wrap our minds around and look at how it might make sense to apply some disinfection to these existing units. The last major challenge we have to overcome with some technology would be the intermittent use of water. So in the middle of the night, we probably don't have much flow. So we don't have to have some disinfection process happening at that moment. So typically we might expect something like 500 gallons per day for a family that one of these is going to treat, but it's really only coming through in some 6 to 8 hour window when people are actually home using the facilities, right? So So there's a few things there that make it so that our typical design or typical disinfection we think about from large-scale treatment plant just are not necessarily good fits. And just to highlight that a little bit of our data here, we have turbidity data from 2 different ATUs. One of them, number one, you kind of see what we know about it is that it's well operated, well maintained, and it's has a small volume usage. There's not many people in that particular home. The second one is a little bit more of a normal scenario where homeowners probably don't know a whole lot about the current status, it's operating, and you see these green bars here are much higher in turbidity, and there's more people using it. The conditions will probably be a little more variable. So when we see that coming out in our data, how many particles, how much stuff would scatter light, that's what we're measuring here. So it's kind of how murky is that wastewater coming out. So that kind of illustrates our challenge here. For example, if we apply UV light, this stuff scatters the light, and the bacteria could even be hiding behind the particles or be shaded from these particles. So we want to apply more light if we have high turbidity, or ideally not have such high turbidity in the first place. So that would be like I said, something that's a little out of our scope, but something that's related and could be improved. Okay, so the first best question to ask ourselves is, well, why not use something that's already existing? Why not chlorine, for example? Why not use our UV lamps that are already using wastewater treatment? Well, that would be nice to keep it simple. We might not get a grant if it was that simple, and so there's some incentive as an academic to try to do something fancy, right? But truth be told, these would already be happening if these conventional systems work well. Chlorine, and as I understand it, most of these systems have the option to chlorine. You could install that, you could use it, but it's not so great when it kills all the little mosquito fish that are keeping your ditch clean, or all the plants and other stuff around, right? So we don't want to chlorinate actually, because chlorine itself is toxic, and in killing the bacteria, you're also going to be killing the natural stuff that's also contributing to this treatment to an extent, right off the outfall. So it would be better to have living mosquito fish and not chlorinate in some sense. Now, I say better, but you still have the pathogen problem, right, if you're not disinfecting. So the other concept might be a low-pressure mercury lamp, which is pretty commonly used for germicidal stuff. They're pretty fragile, they contain mercury, and we have to shield them from the water or the environment with fragile cords. So those are not great options either. And you'd be running them all day long every day. Those ones you can't turn on and off. It's a little bit like our fluorescent lights. If you want them to be on in full power, you have to give them 10-15 minutes to actually pull them up. We might not notice that too much in the pop the lights on, but they actually do. If you design them for a specific intensity, it's not so reliable. And so there's a couple of reasons there why I think our fancy new UV LEDs may provide a good solution. Okay, so just a bit about the technology. I wanted to kind of tell you a little bit about that process. In our lab, maybe we tinker with something, we think that we have some idea that can save the world, can kill the microbes. Once we have that idea, we're supposed to go and disclose that technology formally with LSU to say, hey, here's our technology we're looking forward to applying for a patent, to eventually license this, commercialize it, all of that. That's a big long process. I'll show you a little bit about that timeline in a moment. But that's kind of why at the moment I can show you that we're setting up some things. We've got this nice fancy box now that we're planning to take to the field. The box itself is not the technology, right? That's not what a device would look like anyway. But what is neat is that we're at the point now where we are almost ready to take something into the field, plug it into an ATU, and let it disinfect with a variety of different options. We've mentioned we might combine that with chlorine, maybe we'll combine it with ozone or some other disinfectant. And that's kind of where the technology is coming in, where how does that actually work? And I'm going to kind of leave that to your imagination for a moment. Again, given the tech disclosure stuff. Okay, another aspect, and Dr. Goodman's already mentioned this, the other neat thing about the project is mentoring students. And as much as we'd like to be innovating really, really fast, one of our main priorities here is to educate the next generation of leaders, innovators, and so we've got a couple students here, Maddie and Evan. Evan's a master's student on this project, and Maddie got a chance to help out a bit this summer, and they're there working to assemble this little case that we can use to test our technologies with. So that's been a big part of it, and I'll say, as we look at our timeline, we'll see that that's actually been a big focus of our energy and time this year since starting. Really, we hired Evan in January, and he's already doing great work with us on this. So, the timeline, we all want to be already here cleaning up the waterways, right? That's kind of what I expect is our hope. But first, we have to identify the problem in the first place, right? I was not even aware of the scale or the scope of the ATU systems until 2 or 3 years ago when we reached out to Brady and Ronnie about a potential idea to see if they have some sort of collaboration. Then we learned about it. So us in our ivory tower just didn't even know what the issues are, right? So we've been, others before have been identifying this problem from a long time ago. We're a little newer to that. So then we start saying, okay, well, what could we do about it? Some of those ideas, brainstorming, start testing things. And some of the testing has been going on for some time because way before I even knew about this project, I've been tinkering on other projects with these UV LEDs and disinfectant systems. So we've been doing some things, and I'll show you some of the data on that. From there, then we go into that technology disclosure, the patent stuff, how to commercialize. There's a lot of steps there. But really, that all kind of has to happen before we get to actually applying, right? That sounds pretty obvious, but I just kind of wanted to show where we are approximately, in my mind at least, is not quite here yet. We'd love to move at the speed of science, as someone funnily said about a year ago. Actually, the speed of science is pretty slow, so I'm hoping to start moving towards the speed of innovation, which is hopefully a little quicker, right? So we're working on it.
Where are you at with those bars were?
I just put that red bar. That's kind of my feeling of where we're at. We've not done the formal technology disclosure on putting the ideas together yet, and we have not even had them all together to fully test them yet anyway. So that testing is going to happen on an ongoing basis. So maybe we try something else in addition to it. We might change things, but having that technology disclosure kind of triggers the process towards a commercial product. The commercialization could mean we set up a patent and then license that out to some other company that produces similar devices, and then they are selling these in Louisiana. Maybe that means we make a startup and start assembling them with a Louisiana-based company or something. That's all kind of mysterious because we have never done that before, right? So a lot of this aspect is new. I'm just starting to set my sights on what that's going to take, and as the update, I want to share what I understand to be that process before we can really get something. It's also possible that some company that's working in this space will come up with a treatment device. Hopefully we could get there first, or could make a product specifically helpful for our situation, but there's no telling when exactly that's going to happen. Certainly somebody could do it. And so I mentioned this, but a lot of this training of our students, developing that, even developing this little box that we can take to the field to plug in and actually test things with these micro methods, all of that is coming together. It kind of feels, if you're thinking about our last last update, hey, look, this UV stuff. You know, here, hey, look, this UV stuff. Maybe it'll work. But there is progress there. When I prepared a few slides for this, it kind of doesn't feel like it in some sense, but I want to say that I really do think that there is progress. Okay, so I can show a bit of preliminary data. This is all lab scale stuff. We've not done anything truly in the field. And like I said, we've not integrated the whole system yet. But if we want to look at some data, here's some neat data where we had some UV lights. These are our photos, some UV LEDs. Each of these little guys is one LED, shiny. And that is in germicidal range. We have some advantage here over the mercury lamps because we can turn them on and off anytime we want, no problem. And the color, or the wavelength, they shine, is actually a little bit easier to manage compared to the mercury lamps. So we don't have to have quite as fragile of a protective layer, potentially, and the LEDs themselves are less fragile. And you can think about it, right? We put LEDs in our phones, and if you take your phone and smack it against something, it's probably not going to break the lamp, right? If you do that with a mercury bulb of some sort, you can be sweating bullets about how much glass you're about to break, right? So there's some good advantages just in the physicality of it. And we have some E. coli destruction data here. So this is a log plot where every -1, so -1 is 90%, -2 is 99%. So we can show with this particular system, we're getting 99.9999% E. coli removal in a matter of several minutes. And that's going to depend on how much light we shine, right? We can shine less and adapt. For example, if we had slower flow, if we have some way to adapt to that. We don't need to kill them quite as fast. They're hanging out there a little longer because the water's not flowing fast. Or of course, if we just give them no light at all, none of the E. coli are dying. So that's pretty neat. The other piece I'll say, and this is actually data that we have from a different project, but it was part of our inspiration here, was simply that if we take some different disinfection strategies, sometimes bacteria, in this case our E. coli, can repair themselves afterwards. There's a known thing from UV light. If you disinfect with UV light and then give the bacteria a bit of sunlight or visible light, that can actually trigger a repair mechanism. So one of our questions was, well, can we cancel that out? Can we prevent that? How can we deal with that? So on the left, I'm showing you what happens if we just disinfect with a bit of chlorine and then give them this repair. They don't repair so well after chlorine, but we didn't kill them too strongly with this little small dose of chlorine that I'm showing you. So that killed maybe 40% of them, so 60% are surviving, and they could kind of care less about the light afterwards. But now if we disinfect thoroughly with the UV, and then we can see they can repair. So if it's just from the UV, these little gray triangles, they actually repair a fair amount with some light afterwards. Now, if it's really intense light, that kind of knocks them out a little better. So right in the middle of the day might be all right, but if it discharges in the evening, we get a bit of light or repair. You've got some living bacteria out there that you thought were dead. One thing we could do though is we could add in just a little bit of chlorine. The same amount we used for that. It was only going to kill maybe 30% of them. The UV is doing most of the work, and then in the chlorine base, it's kind of preventing them from repairing at all. So it's a neat little thing that we can do. We can compare that with UV followed by chlorine, and that doesn't work so well. We can do chlorine first and then UV. That also doesn't work super well, but doing them at the same time is a particular advantage that some of our data has shown. So that was pretty neat. That's the type of stuff we're going to try to implement into technology. Like I said, maybe it's hydrogen peroxide, maybe we'll generate ozone, something like that. Put together into a package that hopefully we could put at the tail end of one of our HU systems. So that's where we're at. This is why we are thinking it could be a good idea. Happy to take some questions. I'm sure Karen will be welcome.
So what type of removal will you get with the LEDs?
Well, we haven't tested them in real wastewater yet, or in a flow-through system yet. So the type of removal that we're getting is, we can remove all of them if we give them enough light. There's a question about the rate here. At what rate are they being destroyed? And then we need to calibrate that with the size of the reactor, make sure the water spends enough time in there. So I can't even, I mean, I might be able to do some math to predict something, but with the wastewater, you know, there's a lot of moving parts. We need a few more pieces before we can say. One thing I will say though is, with any of these, you know, this is the 90% removal line. This is 99% removal line. So in a matter of just a few minutes with the intense light, we're at, you know, this would be probably the 5-minute mark, and that's 99.99%. So if we calibrate it correctly, it's not hard to get the right treatment. There are some questions about how much turbidity gets in the way of that, and, you know, in a realistic manner, is that water flowing too fast to get that amount of time, or how to adjust for that?
I have a question, actually, for Dr. Bevins. That the continuous sampler that you were talking about,
Passive sampling.
passive sampler. If you have it out there like that, won't the microbial grow on there? In other words, you'd be getting a higher reading than what's actually is in the water?
Yeah, so that's something we've kind of got to sort through. Right now, we're only leaving them, this is all just pilot, it's us kind of tinkering. But right now, we're only leaving them in place for 4 or 5 days. So we're really not expecting much growth, because typically, you know, the time scales to establish a biofilm and things like that, to get like sustained growth going, are probably on the order of like weeks. So what we're doing right now with them is, I've got an undergraduate student who is, we're actually taking samples from the Natalny, dropping these samplers in, and we're assessing like how much suspended solids they take up over time. So we're trying to get a feel for like, okay, if we leave these in place for 2 days, how much of this suspended material in the river are we going to absorb in that time? And then based on that information, then when we put them in place, we can kind of make some more inferences around, okay, when we see this number on this sample, is that like what it's been taking up, or is that, is it already at equilibrium? Is it growing some, or is it losing some? Those are all, this is like brand new stuff. So those are all to be determined, for sure.
When you talk about the intensity of the light, is that, I know last time you talked about the wavelengths and you did contamination, is that what you mean by the intensity, like the different wavelengths of the light?
That's a great question. And the terms get pretty tricky. So this is not the wavelength this time. This is how much power output, so how bright it is. So this is more like brightness, whereas the wavelength, you could say the one color might be more intense or more higher energy than the other, but this is just
Okay,
right.
so you're not meaning the energy though, it's just like the brightness of it is.
Well, okay, so I'll go into a little bit of the physics of photons for you. So if you have a, we say that photons act like a particle and a wave, and if you have a photon that's doing this, it's pretty high energy, right?
Yeah.
Enough to hit something. So we'll say that photon is high energy, and it took a lot of power to produce that photon. But when we talk about like the brightness or intensity, it's how many photons are there. So we have two things, right? Like you could take sunlight and shade it really well, and it's not very bright, but it has a full spectrum of colors, and we have UV light in it as well, which is at higher intensity. So there's the two aspects. It's like the color can be more energetic, like X-rays are really, really energetic, and then you have like how many photons are actually there, not